Hello, good afternoon. Lower this down just a little bit. All right, good afternoon, everybody. I hope you had an excellent lunch. You're feeling recharged and refreshed, ready to start a double session here this afternoon to learn about the Darwin Core Data Package. Oh, I just need to close that really quick. So hopefully you didn't just stumble in here by accident. Hopefully you are here to actually join us today. If you did stumble in, please stay. What we are going to do over the course of not just this session, but the next two sessions today is bring forth an excellent group of speakers to introduce the Darwin Core Data Package. This first session will introduce the generalities and the general concepts of the data package demonstrated through a series of use cases. Our second session this afternoon will then give you a little bit more of the nuts and bolts and nitty-gritty information that you would be interested in. But before we start, I'd like to take a few minutes to help orient you with some helpful information for today. And so the first thing to know is what exactly is the Darwin Core Data Package? And I'm sure there's a few of you in here who are already aware of what it is. But for those of you who are not, in a nutshell, the Darwin Core Data Package is a publishing model that enables publishers to share richer biodiversity data than what is currently possible with the Darwin Core Archives. So the Darwin Core Data Package mostly utilizes terms from the Darwin Core standard, but it also includes terms that are governed by organizations outside of TADWIG. And I'm not going to linger overly long on this slide here, but just to ease any immediate concerns or anxiety over the potential of learning a new data model and what that might look like, let's just take a quick look at what the Darwin Core Data Package is relative to the Darwin Core Archives. So what do they have in common? And so both of these data models have an associated guide. They include a data set metadata as an EML.xml file, and are data shared as either a CSV or TSV. But where biodiversity data shared in the Darwin Core Archive format are confined by cores and extensions and limited by the star schema, the Darwin Core Data Package does not have those limitations. And that means that the Darwin Core Data Package can support data that are much more complex and that cannot currently be supported, including organism interactions, including identifications that support multiple taxa, and so forth. Now, let's have a very quick look at our agenda for today. And the first of our two sessions will help you visualize the practical implementation of the Darwin Core Data Package with 5 use cases. And we ask that you hold your questions until the end of the session. So we will go through all of the use cases back to back and then have time at the end to entertain any questions that you have from there. At 3:30, we will have a 30-minute coffee break. So everybody can go out and stretch your legs, get some much needed caffeine, and then hopefully you'll come back at 4 o'clock, straight back to this room, join us here to listen to another 5 presentations addressing many of the practicalities associated with the data package. In the first presentation, then we'll provide a solid overview of how the data package came about, drawing on the use cases presented here in the first session. Subsequent presentations will provide an explanation of the relationship between ABCD in the data package, current implementation status of the data package at GBIF, and insight into the data package publishing tools that are currently in development or being considered for development. And then finally, you'll learn more about how you can get involved in the current public review process before we close out the symposium with a Q&A session. All right, so there's some final information that you definitely want to know. Timing me. Oh, okay, thanks. I'm getting timed. There's some information you're definitely going to want to know as we move into these upcoming presentations. And this is going to be all about what you're going to see in the represented publishing models used to implement the Darwin Core Data Package. In this schematic, there are 6 components. So the first component are represented as events, right? Events, just as in Darwin Core Archives, are things that happen in a particular place in time. Next, you're going to see what are called entities. So entities include essentially interesting information about an event or something that happened. The third component you'll see are called agents. So agents are the things that engage in an activity or an event. An agent can be a person, a group of people, or an organization, or even AI. Next, we have join tables. And join tables are exactly what they say they are. They are tables that join information about what happened to the interesting things and the people or in groups that did them. Okay, so with these publishing schematics, you will also see arrows. And so these arrows provide directionalities. They link the tables and show you how those tables are linking to one another. And then finally, you're going to see what are called predicates. And predicates are the words or phrases associated with the arrows. These are words or phrases that describe the relationships between those tables and relative to the directionality of the arrows. Hopefully that's clear, translucently clear, maybe not crystal. Yeah, excellent. All right. Perfect. Okay, so bringing it all together in a very simple example, I'm probably running out of time, but we're going to continue anyway. Consider a situation where you have an occurrence with some additional information for which there are no specific Darwin Core terms. In a Darwin Core archive, you would have an occurrence core table, and you would have an eMOF extension to attach that extra information. As a Darwin Core data package, this same information would be shared as follows. You would have an inferred occurrence that occurred during some event. Then the additional information about that occurrence would be connected to the occurrence using an occurrence assertion. So in this case, an assertion in the Darwin Core data package is the equivalent, is attaching the equivalent information that you would once use in the eMOF extension in the Darwin Core archives. And without further ado, I would like to say, let's go ahead and get started with sharing our presentations before anybody thinks of any questions to interrupt. And I would like to invite our first speaker, Carrie Seltzer, who will be presenting. Oh, your presentation's not up. There you go. Presenting on a data use example from iNaturalist.
Thanks, Kate. My name is Carrie Seltzer. I'm the head of engagement for iNaturalist, and I'm just going to stall for a moment while my slides are pulled up, I hope. And great. So I'm going to be showing you how this Darwin Core data package maps to iNaturalist occurrence records. I also want to give a huge thanks to John, who was tremendously helpful in helping me understand how iNaturalist data maps to the data package. So first of all, if you're not familiar with iNaturalist, iNat crowdsources observations and identifications from all over the world, and it's primarily photo-based. These are occurrence records, and occasionally there are also organism interactions associated with those occurrence records. Sharing biodiversity data is a huge part of iNaturalist's mission. And so we share a massive data set with GBIF. And I am pleased to highlight that if you look at data since 2020, more than half of species have more than half of their data from iNaturalist. So pleased to share how this would work. Just to recap, for those of you who maybe, actually, let me ask, who uses iNaturalist? Okay, cool. If you have not used iNaturalist, here's how it works. You see something, you take a photo, like a dragonfly, for example. You trust, and at that point, then people can either confirm the identification if you've submitted an identification of your own, or they can advance it or correct it if you've made a mistake. Mistakes happen all the time on iNaturalist, but they can always be corrected by the community. So keep this flow in mind. A person takes a photo of a thing, posts it to iNaturalist, and then other people chime in and help with the identification process, because we're going to come back to that and how it maps to the data package. So right now we're sharing a Darwin Core archive with GBIF. We update it once a week, hopefully. And in terms of some of the limitations right now, we are not, for example, passing along all of the identifiers who've contributed to an occurrence record. We're only passing along the first one who, the first person to essentially like put that species level identification. Interactions are captured in iNaturalist occasionally. It's not part of our core data model, but this can be mapped to the Darwin Core Archive format using the resource relationship, but that's not specific to interactions. It would sort of fit interactions into a more generalizable structure there. And lastly, there's a lot of additional data that we call annotations in iNaturalist. It's things like life stage or plant phenology stage. And right now, a lot of those are getting mapped to the dynamic properties field. For example, leaf phenology ends up there because there's not a more structured way to share that in the Darwin Core Archive format. So people are at the center of iNaturalist. And so we're going to start this mapping process with the agent, which, if you have made an iNaturalist observation, is you. So the event is you see something that you want to take a picture of. So now we've got you, the event of you seeing it, and the thing that you saw. You take a photo that is captured in the media. Then there's an identification, and it can come from you, or it could come from someone else, many other people. And then there's that extra data, like life stage or plant phenology. And with all of the records on iNaturalist, as well as all of the media, there is a usage policy. We use the Creative Commons licenses, so that fits in here too. And we've got the provenance. Where did these things come from? So now when we start to put those into this, we see that there was an event conducted by a person. And in this case, it was you there's not sampling effort. It's just you saw a thing and you took a photo. And that the identification of what you saw is based on the media. The identification is also mapped to an agent, which, like I said, can be you or someone else. And the occurrence assertion is being made about the occurrence, which happened during the event. We're just going to keep adding more arrows and boxes, so brace yourselves. All right, so now we've got these join tables in here. Those are the ones in yellow. So now, when it comes to media, we've got media both about the event media about the occurrence media about the occurrence. We've got the media usage policy, which connects the media with the usage policy. We've got provenance media connecting those two boxes. So this is sort of the core, this is how most of the iNaturalist data that we are currently sharing through the Darwin Core Archive would get mapped to the data package. And like I said, we've also got these interactions sometimes. So now that comes in, and when we add the tables, the additional arrows and boxes to that, we've got media that is about this organism interaction, and the organism interaction is happening with or by an occurrence. I know that's a lot to take in. If you want to see this sort of captured in sort of a tabular format, these are the key the key tables that are included. There's some links in the slides here if you'd like to refer to the example data set that John prepared. I forgot to mention that from the outset, that was part of this exercise, was taking a very small subset of iNaturalist data that happened to have that additional information about, in this case, plant-animal interactions, that was used to go through this exercise of mapping iNaturalist data to the data package. So when it comes to iNaturalist data using this new format, there are many new possibilities. For example, one question that we get fairly often is, can projects be represented in the data that is shared with GBIF? And this, for example, could be part of provenance. So I think it opens up a new avenue for us to pass on information that people have been asking for in this more elaborate data model. Another interesting possibility is that the community ID, which is essentially an algorithmic thing on iNaturalist, it's sort of a, the shorthand is there needs to be at least 2/3 agreement at the species level identification for something to become research grade and get shared with GBIF. And so that sort of simple algorithm could, for example, be an agent in this model if we wanted to sort of build out the complexity of information that we're passing from iNaturalist to a data aggregator like GBIF. But when it comes to thinking about where we are now using the Darwin Core Archive to the implementation process, we will need to think about a lot of costs, including complexity and processing time, and what are the resulting file sizes and sharing this way. It currently takes us about a week to put our Darwin Core Archive, and we do it once a week, so it's a constant process. If you've ever found it out of date, it's because it got too big and we needed to optimize again. So with that, I am going to say thank you and turn it over to the next example.
Thank you, Carrie, for that awesome example of citizen science data being mapped to the Darwin Core Data Package. Our next speaker, Jörg Holicek, will be providing an example using natural history museum collections, so probably something quite familiar to everybody here.
Yeah, hello. I'm Jörg Holicek from Botanic Museum in Berlin, and I'm working at the Center for Biodiversity Informatics and Collection Data Integration. Yeah. And I've been responsible for pushing our data for the last years to GBIF and likewise biodiversity networks. So I knew a lot about the limitations of Darwin Core, and yeah, so we were happy to be one of the use cases to publish natural history collection data as Darwin Core packages. So the vivarium we have in Berlin is the largest one in Germany. Right now, there's about 4 million specimens. It has been largely destroyed during the World War, but it grew very fast again due to the distribution of duplicates. And even now, it's growing constantly with about 30,000 new specimens per year, mainly through donations from botanists or by collection events from our colleagues. The type of data we provide, or we are able to provide, are of course the specimens, currently about 4 million ones. About 10% of them are digitized so far as high resolution images. They're stored as TIFF data, and we provide them as JPEGs for the creation of thumbnails, and of course through image viewers for humans to be able to zoom into different areas of the image and to take measurements and to rotate, stuff like that. We do have 30,000 images of the organisms before being collected. Often that's in the original habitat, so that the surrounding flora is also captured. And we do have 30,000 tissue samples and 50,000 DNA samples in our DNA repository. We do provide the sequences through eDNA and NCBI with nightly updates. So far, so before Darwin Core Data Package, we provided only the specimen and the specimen image to GBIF. The tissue and the material samples we shared with the Global Genome Biodiversity Network, because this network also is able to show all the associations between the objects. So you can, if you're looking for a DNA sample, you can go to GGBN, find the DNA sample, and then it will link you to the tissue sample this DNA has been taken from, and then to the specimen that the tissue has been taken from. And as I said before, the sequences are shared with NCBI. Now again, publishing model, as Carrie showed. Again, of course, a specimen documents the existence of an organism. An event documents the gathering event documents the existence of an occurrence at a given location, of course, and then all the materials that get sampled or that get gathered, of course. First is the specimen, that is the specimen gathered on site. Later, the material sample, the tissue sample is taken from the specimen, and the DNA is taken from the tissue sample. These are the arrows. They show that this chain of derivation, and the arrow that I just added is the only exception. We have some tissue samples that are not taken from specimen. For example, if the tissues are taken directly in the garden, there's no specimen gathered, so these tissue samples are derived directly from the organism in the garden. The population of specimens is based, of course, on the material, on the specimen, with the exception then if no specimen has been gathered, then the identification is, of course, based on the organism in the nature. And we do have, of course, a lot of material identifiers. So these are globally unique and permanent identifiers for both the specimens and the tissue and the DNA samples. They resolve. So if you use them as a script, you get the metadata on these objects as JSON. And if you're a human using a browser, then you will get the same metadata as a landing page, and of course, the image shown. We provide certain material assertions on DNA samples, for example. And we, of course, as mentioned, we do have images of the specimens as occurrence media and of the specimens as material media, and of the occurrences. These are the images of the living organisms. The lessons we learned, we could provide way more data than we could before. And with this, there was also a lot of more data issues to be discovered. I learned a lot about control characters that can break CSV and the IPT. We found almost every special character or control character in our data, and we had to clean them. Of course, we had to do a lot of juggling because the data model of our database is very normalized. So I had to concatenate a lot of tables and create a lot of SQL statements for that. And as I showed in the example, there are a lot of objects that don't stick to the rules, and this was just one example. And we had to build a lot of workarounds for these special cases that don't stick to the regular schema. So together, it was a lot of juggling, but in the end, everything fit in, and we were able to provide way more data than before. Yeah, thank you.
Thank you very much, Jared. I'd like to invite our next speaker up, Yingming Gan. She's going to give you an example of the data package with complex marine survey data. And we are actually quite a bit ahead of schedule. Everybody's been super efficient in their presentations. So we'll keep moving through our use cases, but this will just give us a little bit more time at the end for discussion. So continue to hold on to your questions and comments, and we will address those at the end, but we'll just have a little bit more time.
Oh, Thank you, Kate. Thank you for inviting me. Thank you for being here. Good afternoon, everyone. I'm Ming from Scott and Topic Biodiversity Portal, which is also a GBIF and OBIS node. So I'm very excited to share with you how we use Darwin Core Data Package to map our complex marine survey data sets. The Broadwest fish data sets contains fish records from survey conducted from January to March 2006 in the region in the Southern Ocean. The goal is to have a baseline assessment of the biodiversity in that area. It was published in year 2008 as a single occurrence table. Now we wonder how much more enrichment of this data set can be done if we map it to Darwin Core Data Package. So first of all, we have the events, which consists of either the rectangular midwater trawl routine sampling, which is the first 200 meters, target sampling, which is detecting the assemblage and low the net to the depth of this assemblage and sample the assemblage. So we have a survey target that includes target taxon, life stage, and body size for this routine or target pool, which happen during an event. And this sampling contains measurements such as the depth, not open size of the net, as well as the trawl distance, speed, and sample volume. And this goes to the event assertion. All the fish sample the occurrence, and we can also indicate whether the occurrence satisfied a survey target. And all of the fish were identified, and the identification goes to the identification table. Aside from the group of fish as an occurrence, each of the individual fish were also preserved as material. And their weight, so they contains the dry weight and wet weight, as well as standard length and energy content. This goes to material assertion table. They are also photographed. Each of the individual fish were photographed, so we have the of the material. And here to build on the tables, the materials goes here. There are material of an event which serves as an evidence for an occurrence, and it contains also the identification as well as the material assertion. For some of the fish, their stomach content was also assessed using binoculars. The prey item were identified and counts, and the degree of digestion were recorded. So to do this, the prey items and its identification goes to the material table as a nested material. Degree of digestion is accession to this material. Now, it was quite complicated tables. In total, we have 3 tables, which is a lot. It cannot fit into all the slides. And we had a thought exercise. Can we map all this with just Darwin Core Archive? So we could do most of the things with Darwin Core Archive. For example, the events can still be an event core. Survey target and survey were originated from the Humboldt extension. They got flattened into a single table as Humboldt extension. Occurrence, material, identification, and media can go to the occurrence extension. Material assertion and event assertion can go to external measurement or fact extension that points to both event core or occurrence. Now, this is where we felt Darwin could not represent our data set well. First of all, there's less freedom in specifying survey target. We want to indicate a maximum body size for the survey target, and we can put that as verbatim target scope, but it's more difficult to parse. And then to us, we feel because the individual fish has a material entity ID. And for occurrence extension, we use occurrence ID to represent each row. And using occurrence ID to represent material feels a little awkward. The items were published because the media is extension too far. We can use associated media, but that will miss the metadata, such as the license of the photo. And the prey items, we find that it's difficult to represent in occurrence extension. So these are the lessons that we learn. During the mapping, I found that we have to join tables to get the foreign key, just to split them into multiple Darwin Core Data Package tables. And we find that tools for splitting tables would be very helpful. Secondly, we think that it would be extremely difficult to maintain the identifiers upon updating the data set. There are 23 tables, and there are a lot of identifiers to maintain. And we think the Darwin Core Data Package is extremely helpful to model complex data sets because it preserves the of the data represented closer to the reality and preserve the meaning of identifier as well. And finally, I want to go one step further to see if we can access the data from Darwin Core Data Package. A first use case we could think of is the infer detections. This is what humble extension intended to do. Shell. So for example, we have a target scope of species ABC. Say we report detections of an event, including all the targets that we report that we detected. So if we detect species C and we report everything, we can infer that species A and B were not detected. And anything we detect outside of the target scope are the non-target taxa. So using this concept, I'm looking at these 4 tables, looking at occurrence. In this data set, occurrence were reported. So looking at the occurrence of all of the events that are not, that satisfy the survey targets can be used to infer what were not present. So the y-axis are all the events, and x-axis are all the target scope. So we can see the pale blue are the detections from these detections only data sets. The second use case that we thought of is we wanted to be able to pull this data out for data products. So in essential ocean variables, the fish length frequency distribution is one of the sub-variables. So we want to see if we can pull out the fish length and build this fish size frequency diagram. So we look at the material assertion, where the fish lengths are, and then look at how each trawl type it is from, whether it's from routine or target trawl. That's information in the survey table. So the answer is yes, we can do that. We can plot out a fish length frequency distribution. So during this process, I also think, fish length frequency, we put it as material assertion, but it could also be an occurrence assertion. One of the examples from the OBCUSA data set here is the diver could use this tool here to measure the fish while swimming and estimate the fish length. So what I think is important is that when aggregator aggregate these data sets, the variables can come from multiple classes. So fish length can be both occurrence assertion or material assertion. We think this is important to take into account so that user can pull this data out. And finally, I would also like to see what we can pull out the diet composition from the Darwin Core Data Package. And yes, it is possible we look at the diet composition of Pleurogamma antarcticum from both routine sample from a routine tool or the target tool. And these are what we found. And all this was not possible because the data was unpublished. So I would like to thank this list of people who helped me so much in this process. And of course, this data set was a legacy data set. There are mistakes, there are things that are incomplete. And I do this as a data manager and not an ecologist. So there will be mistakes. And finally, this mapping is part of the contribution to the OBIS data coordination group. The coordination group is open to everyone. If you would like to involve in this conversation with both GBIF and OBIS, please, I would like to invite you to join the coordination group. And all this is not possible without fundings from BELSPO, and we are very grateful for that. And thank you so much.
Thank you very much for that, Ming. And I'm trying to look out at the audience and see how everybody's feeling, kind of gauging, is everybody still feeling all right? Not feeling too queasy about excellent. I see some thumbs up. This is great to hear. So three use cases in, we're getting a little bit more complex, maybe a lot more complex with some of that marine data. We have 2 more use cases to go. Both of these are virtual presentations. So presentation number 4 is by Cecilia Svenningsson, and she is presenting a use case that includes both citizen science and DNA metabarcoding data.
Svenningsson, and I will present how my co-authors and I mapped a citizen science and DNA metabarcoding survey to Darwin Core Data Package. The data behind the Darwin Core Data Package mapping comes from a national survey of flying insect diversity across 5 different land covers. The project was called Insect Mobility. Insect sampling was carried out by volunteers that drove on pre-designed 5-kilometer routes with large nets mounted on the rooftop of their car. The end of the net could be detached from the larger net after sampling, and the contents shipped to the National History Museum of Denmark for identification. The project consisted of a survey with individual sampling events for which there was collected material. Some of the material was photographed, and identifications were based on both morphology and DNA analysis. We've previously shared the data as a Darwin Core archive, but because of the star schema limitation, it was never really a good fit, and we wanted to see if data could be better shared with Darwin Core Data Package. So if we go back to the Darwin Core archive, in the ideal world, with the table options we have in Darwin Core archive, we would first share the sampling events, and from those events, we collected the material. We took images of that material, we measured biomass of the bulk sample, and we extracted DNA. Occurrences was based on both the DNA derived and the morphological identifications. However, this would not work in a Darwin Core archive because we have both extended information, the event and the occurrence table. And in a Darwin Core archive, you can only have one core file, either an event, occurrence, or a taxon, and only one layer of extensions to the core file. So the sampling event data had to be shared as occurrence core in order to include the DNA-derived data extension. This meant that media, event information, extended measurements of facts, and part of the DNA-derived information was shared for each individual occurrence. Example, an occurrence record of a growth beetle in GBIF includes an image of the bulk sample the species was detected in, and not the individual itself. And we had several images taken for each bulk sample, so you can imagine how the media quickly got inflated and did not represent the organism that was detected in the occurrence record. So how would this data look like in Darwin Core Data Package, where we are not restricted by the star schema? I'll now go through the tables we included in the Darwin Core Data Package. Some boxes or tables have dotted lines around them. This means that they are not actual tables included in the data package, but the information is included in the tables they relate to. Our citizen scientists were agents that conducted surveys. Each individual survey had a specific land cover and taxa target. The agents followed a sampling protocol to carry out the individual sampling events. During each sampling event, material was collected. The material was bulk insect samples, and images were taken for some of those samples. The bulk insect samples were processed into two size fractions, and dry weight biomass of the size sorted material and DNA concentration of the purified DNA extracts was captured as material assertions. From the bulk sample, we performed morphological identification of certain taxonomic groups. We then carried out nucleotide analysis with 3 different primer pairs on a fraction of the purified DNA extract by following a molecular protocol. And from one of those primer pairs, we shared sequences for which we compared to a reference database to get taxonomic identifications. So we had identifications based on both morphology and DNA sequences for which occurrences could be inferred, which were then evidence of the occurrence of taxa in an event. And the occurrences satisfied the survey targets we set. In total, we mapped data to 12 tables in Darwin Core Data Package. We specified the preferred identification, the DNA sequence, with the is-accepted identification term. We could elaborate on how material was processed by including the part of and derived from terms in the materials table. The quick reference guide and the relationship explorer was a great help to understand which tables would make sense to prepare first in order to have the identifiers needed to link between the tables. So we went from a Darwin Core archive with a core file of 4 extensions to 12 tables in a Darwin Core data package. If you're curious about which terms and content we put into the individual tables, you can find the use case example in Sigmobilen under the survey folder in the Darwin Core Data Package examples repo. We found the Darwin Core Data Package enabled us to establish the right links between the data from the project and share even more information than what was previously been shared in a Darwin Core archive. So Darwin Core Data Package is definitely a better fit for DNA metabarcoding survey data. Thank you all for your attention, and I hope you have an excellent time in Bogota.
So our final presenter, Holly Little, was unable to join us here in person today as planned due to the government shutdown in the US, but she was able to submit her presentation as a virtual presentation, and I do believe she has colleagues online to answer questions when we get to the Q&A session at the end. But I would like to now present to you Holly Little's presentation. She's presenting to you today a use case on paleo data collections. Technical difficulties.
I'm Holly Little, Informatics Manager in the Department of Paleobiology at the Smithsonian Natural History. With my co-authors, I'm here to present a Darwin Core Data Package use case for fossil specimens, or as titled, Characterizing Paleontological Specimens for Maximal Alignment with Global Data Practices. Informatics Manager in the Department of Paleobiology at the Smithsonian National Museum of Natural History. With my co-authors, I'm here to present a Darwin Core Data Package use case for fossil specimens, or as titled, Characterizing Paleontological Specimens for Maximal Alignment with Global Data Practices. Our use case addresses the mobilization of data derived from specimens in paleontological collections. These collections cover evidence of all types of life on Earth through time, and key data points include documentation of that material, the representation of taxonomic occurrences through that material, associated media, and critically, the geological context, collections event, and location information for that material. Often, fossil specimens also include important relationships, including biological interactions and material associations. Our overarching goal is to improve the discoverability of paleo collections data, which is largely dependent on the details listed here. Findability and interoperability of key data points, ensuring there is clarity about what the record represents in the physical collection, being able to share the variety of associations or relationships you might find with fossil specimens, and having more options for fossil data because of the variety and complexity of data requirements. For more context on the scope and complexity, here's a brief, highly selective, and not comprehensive view of that variety in fossils. As noted, paleo collections cover the fossil equivalent of all modern biodiversity groups. That includes plants, invertebrates, vertebrates, trace fossils, etc. A fossil specimen in a collection could be a single microscopic organism like this foram, a jar of them, a single slide with thousands of fossil pollen grains, a stratigraphic sample of dozens of mollusks or a single mollusk, a slab from the size of your hand to the size of a treetop with many organisms on it, a drill core from across many stratigraphic layers, and of course, large, full skeletons that might be a single organism or a composite of many. Somewhat independent of the use case and testing for the Darwin Core Data Package development, we determined it was necessary to try to categorize the wide variety of fossil specimens to better understand what the various types of fossils we manage in the paleo collections are, and to more clearly define what the necessary data structures are for each type. We worked closely with the paleo community through the Paleo Data Working Group to complete this evaluation. Our working sheet is shown in the screenshot here. This effort resulted in 3 primary data groups that inform the data structure needed. Each data group covers many different types of fossils as managed in paleo collections. Group 1, standalone record, covers the most typical data structure needed when a fossil specimen can be represented by a single record and most commonly a single taxon. Group 2, relationships, delves into requirements for clearly defining associations, either biological or material, between individual taxonomic occurrences contained within a single object or sample of multiple objects. Group 3, complex stratigraphy, addresses fossil samples that may have multiple geological contexts. This is most often core samples. That work has also helped us to develop values that can be used to further develop a vocabulary for a newer Darwin Core term, material entity type, which is key for describing what a fossil actually is. Paleo collections have, of course, been publishing data mapped to Darwin Core using the Darwin Core Archive model as long as it has existed. But that version does have limitations for meeting our data publishing goals. Events and localities are critical for paleo research. While that data can be shared with the Darwin Core Archive, it often has limited discoverability. Importantly, in previous published data, it is actually quite difficult to know what exactly that record is documenting. For example, if you're looking at a single record for a fossil insect, you might not know if that record represents insects on a slab, as shown in the top picture here, or insects in a piece of amber, as shown in the bottom picture. It also might not be clear that all of these individual insects and one spider of a variety of taxa are often found on the same object in either case. In addition, previous models have had limited flexibility for using data structures more tailored to the different types of fossil specimens with variable specimen data. Before diving into our mappings, we wanted to share a few caveats. Because the Darwin Core Data Package is much more dynamic, we manually created our test data sets record by record and term by term. We're still refining those test data sets and piloting various iterations for each data group. And it is quite possible we will define more data groups as we go. I'll also note that much of what we have determined is likely adaptable for modern or extant biological collections as well, since many of the data requirements might be shared depending on the type of collection. So to start, a major shift in the updates to Darwin Core and the Darwin Core Data Package is use of material as the primary class for collection specimen data. This change introduces improved options for clearly describing the physical object represented by the digital record, and an increased focus on events improves discoverability of this class of information and the connected geological context terms. Expanded tables for capturing agent data allows us to share clear information about people associated with our collections or shared across collections, which is common for paleo. This part of the mapping is pretty much shared across the data groups, so for the rest of the diagrams, we selectively remove some tables for visual simplicity and clearer focus on the more unique aspects of each profile. As we focus in on data group 1 and some versions of data group 2A, the material relationship, we expand into more tables. In our testing, we found a few key areas that needed particular attention and worked with the Darwin Core Data Package team to address our requirements. Location is not a real class as it is embedded with an event, but we chose to show it in some form here because of its importance to fossil specimens. There can be a lot of nuance in the relationships between collecting events and locations, with many to one relationships in either direction. Due to those nuances in the event and location data, there can be some variation in how to most clearly describe the geological context. This is addressed by having an ability to link geological context to either event directly to the material. In previous TIWIG meetings, you may have heard our group talk about challenges with fossil taxonomy. Having an ability to mobilize the taxonomy used to build out the published data set with a table-like identification taxon helps to address any issues with aligning two shared taxonomic backbones like the catalog of life. And finally, we can now use terms within the material table to note those physical relationships to other material entities. As with other biological specimens, fossil material entities have biological relationships to other material entities. For data group 2B, biological relationships, we integrate the occurrence table for improved mapping and publishing of data about these types of relationships. In fossils, this can be found in many forms, including traces like insect damage on a plant, a single object that preserves encrusting of one organism on another, as shown in 2 of the images here, and an organism within another organism like these fish. Referring back to the need for clarity about what is physically present in the collection, adding the material relationship back into the biological relationship model enables us to fully describe many types of fossils. In the images here, we see examples of fossil insects, each included in our test data as its own record. They're each evidence of a taxa at a time and place, so an occurrence. And they also all happen to exist on the same piece of shale. Therefore, having a material relationship and shared event, location, and geological context data. The model shifts to a much more event-centered version for group 3. Here we typically have a core or other type of stratigraphic sample collected in one event and at one locality, which could cross multiple stratigraphic layers and therefore have multiple associated geological contexts. That core might be intact as a whole single object, or it may have been processed into multiple subsamples as shown in the 4M slides here, which probably contain multiple individual organisms each. In addition, core samples include material beyond biological. This mapping profile with the Darwin Core Data Package enables publishing of a single data set to make those events and associated material discoverable even if only parts of that data set are relevant to an aggregator like GBIF. In our work so far, we have developed 2 test data sets using these 3 mapping profiles. In total, the test data sets use about 11 tables, but with varying levels of emphasis and complexity depending on the type of fossil. Moving forward, we will integrate additional tables that further enhance these records as we continue testing and evaluation. Given the range of fossil types and challenges in the data, this use of multiple mapping profiles will be essential for fully leveraging the Darwin Core Data Package for fossils from paleo collections. Ultimately, the Darwin Core Data Package does enable better representation of that complexity than previous publishing models. While that is true, it will take work to implement. As I noted in our caveats, we had to map our data manually. Most collections management systems do not have the fields or tools to store or generate some of the necessary data that makes the Darwin Core Data Package a powerful publishing model. In particular, the extensive and important use of identifiers is something our community doesn't quite have the ability to meet yet. And as we all see within our communities, the resources and expertise to adapt to a new system can vary widely across collections and institutions. To help meet the needs of the paleo collections community, we're working with the Paleo Data Working Group to develop a library of shared resources and tools in a new web resource we're calling the Paleo Data Knowledge Hub. We're also working to create a Darwin Core implementation guide that is specific to fossils from paleo collections and will address the Darwin Core Data Package once finalized. We're also working to create example records for the various fossil types and data groups, which can be used to illustrate the full data mobilization pipeline alongside the implementation guide. And with that, thank you. We really appreciate our partners in this process, GBIF and John, of course, and a big shout out to the Paleo Data Working Group. Also, thanks to the organizers for inviting us to contribute to this session. We are more than happy to talk about this use case in more detail and any other details you might be curious about for fossil data. Please reach out with any questions, comments, etc. My email's here, and Erica and Alex are available in the session for any immediate questions you might have. Thanks.
So this concludes the part of our symposium where we present use cases, and I'm actually quite blown away, not only because I feel like all of these presentations were absolutely fantastic, doing a great job, hopefully you think as well, to explain what can be quite difficult to absorb as far as thinking about your data in this new data model context. The other thing I find incredibly impressive is we are 15 minutes ahead of schedule, and I can't remember the last time I was at a conference session where this happened. So congratulations. Thank you very much to all of our speakers. This gives us another 15 minutes for Q&A. So it is currently 3:05. We have 25 minutes for questions, and I'd like to open the floor to anybody out there.
Well, thanks a lot for all the presentation. I really enjoyed the use cases. My question or my comment is more about the usage of the data package as a way to standardize the data. I work for a biodiversity observation network in Quebec in Canada, and we are working to standardize the data of a lot of researchers, or sorry, perhaps I can move a bit. Researchers or our partners have a lot of data, and we often encounter the same challenge, is that their data is very unstructured. They're basically Excel files, and they use some glossaries to like create some unique identifiers. And we found that just standardizing those data set into Darwin Core archives requires a lot of work, and often this is not budgeted or planned. And what I see with the data package is that there is a great opportunity to have more richness, but I have a concern that it will require exponentially more effort to standardize the data. And we still have the challenge of there's not a lot of money or efforts put into that. So I'd love to hear your comments on that effort to standardize and how we can work toward this with this new standard.
Hello. That might be a question better suited for later, but we have so much time, I'm going to take it. And the answer is complex. We already have problems with resources, with mapping Darwin Core Archives. The big message here is that Darwin Core Data Packages enable those who need to do something more than they can do already and have the resources to do it. We all want to find resources to do things more better and richer, and to try to do, if those researchers have more data than they can mobilize via a Darwin Core archive, they have a choice. Find those resources and publish in a Darwin Core archive, or use it, or sorry, in a Darwin Core data package, or do things the way things are now, not include all that richness, and use Darwin Core archives. Both will continue to be supported. Does that help? Okay.
Thanks. So this is just to kind of check where everybody is with this sort of stuff. So the Darwin Core Data Package, I just checked the GitHub and it's quite sprawling, but luckily very formally described apparently in JSON. So I saw in some of the talks, people saying, well, this is really complex to map onto this. I have a SQL database with complex joins. I have my tables. And I'm just curious to hear, would other people do the thing that I would do, which is to load the Darwin Core Data Package description into an LLM and then have it talk me through what I'm supposed to do? Or I mean, this is too big for our own brains, it feels like to me. So I don't know if anybody else is doing the same thing here. Not just me, okay. Cool, thanks.
Just 1 second. We have 2 questions online, so we'll take one of those.
So one question is from Matthias Dillon. He's asking, more tables mean more identifiers, and what should they do with the occurrence IDs they currently have in Darwin Core Archives? Do they want to transform it to Darwin Core data packages? How many more identifiers do they need to mint, and how stable do you keep those identifiers that they have already?
Thank you, Matthias. I hope you can hear me. I think the question would really be, if you're trying to move from Darwin Core Archives into the data package, it will be because you're trying to express more relationships across your data. And in order to do so, you would need to uniquely identify the entities that are in there. And that will require defining what would constitute an ID for the entity that you're creating. So at the moment, an occurrence record quite often flattens a material and the event of the organism existing into one record, and you've got an occurrence ID. It would be quite likely you'd have to identify how you would uniquely identify one of your material records, which would be something that the publisher would have to do. We recognize that tools would need to be created to make this easier for people, because it's going to be hard. And I would expect tool developers, such as the IPT, for example, to have to help people manage these things. I would also expect that to be the case that some of the identifiers will be unique and persist across time, while others might be only valid within the Darwin Core Data Package as it's being created. So we'll have certain things like specimen IDs, which are probably stable across long periods of time, but there could be others that really only need to exist within that version of a data set. And I think we'll probably end up in the situation that we've got two classes of identifiers, some that you can rely on and others that are really just joining data within one data set.
Matt asks that we introduce ourselves.
I'm sorry, I was asked to introduce myself. I'm Tim Robertson. I'm Deputy Director and Head of Informatics at GBIF.
And I was and still am John Wieczorek.
Thank you very much, Eduardo Klein from Integrated Marine Observing System in Australia. Thank you very much for the presentation, especially the use case were very illuminating, if I may say. But I think that from the provider point of view, this new data schema solves the problem of the STAR schema, which is the lack of flexibility, basically. But it creates a very complex structure. It's standardized, could be standardized, but it's complex. Now I'm thinking from the user point of view. The STAR schema presents some blockers for the non-expert user, especially the EMOF with this long format, multiple possibility of adding variables, et cetera, so you need to know how to work with that. This schema is extremely complex for the non-expert user. How do you think it will be accepted in the community? Because for the providers, it's perfect. I mean, it's perfect. And I may say, if I can accommodate my data into the Darwin Core STAR schema, I will prefer to use that. Thank you.
It's complex. But you have to realize it's only as complex as you need it to be also. So you've seen a bunch of diagrams, one with 20-some tables used, one with 6 tables used. It's because they didn't use all 77 tables that you could use. They're all there because the model was built to cover every use case we encountered. So they all had to be there to solve something. That doesn't mean you need it. No. And in fact, you may not need as many tables for your case as was seen there. In fact, you may not need Darwin Core Data Package at all, because the STAR schema isn't really an obstacle to you. This week has been illuminating already, starting with the Global Nodes meeting, where people were doing exercises trying to map into this thing with an artificial use case. From that, I got the feeling that a Darwin Core archive serves as a very good stepping stone to that. And the thing people need to understand is what that can do that the archive can't do. And then how much of that is actually relevant to you. And then make that decision. And it's going to be perfectly fine to stay where you are with Darwin Core Archives for a lot of people. But as we've seen in our community, as people start to get expertise, they start to get a little bit nervous about things that they're not doing what they think could be the best way. So when they get the resources, they learn. And when they get more resources, they do. And I think that's how it's going to go. That we're going to get, as a community, we're going to evolve into using that when necessary. The other side of the getting stuff down. We're going to share that question.
Oh, wow. I feel like I've just been handed This is Tim again. That's a really good question. Because some people will, want to share simple data, and others will share perhaps more complex data. But then you've got users who will want the both kinds of complexity, different ranges. Through our work, we have admittedly been focusing on how to support the publishing community at this point. We have groups that are frustrated that their Darwin Core Archives does not reflect their research. So our focus has really been on that. The challenge, I think, is going to be the next stage for us is to start thinking about what aggregators like GBIF and others would have to do. And we will be moving into that kind of stage now. I would expect an aggregator like GBIF will still need to produce very simple views of data, things like occurrences, things like material catalogs with specimens, probably with relationships across them. But that's already a step forward from what GBIF can do today. We don't have a material catalog. We've just got occurrences. We can't do relationships across material. So I could imagine GBIF would be able to have simple occurrence views, a catalog of material and the relationships, occurrences with the evidence that supports that. Here are the specimens in support of those occurrences. I could imagine GBIF having a catalog specific to the evidence of organism interactions, for example. So simplified views on top of this richer model. We don't know what that would be yet. And at this point, what John and I and the team and everybody has got to is we're trying to share with you through the last few years, we've been working with lots of publishing communities. This is the kind of data that people are sitting on and want to share. And we're trying to socialize that. We've got it to this stage. We've got it to a shape that we think accommodates a lot of use cases. And we want to socialize that with you, with the TADRIG community and the biodiversity community, to know what we should do together to move forward. This is a checkpoint for us. It's a real big milestone to know, people are sitting on this data. You've heard some case studies where they've expressed, this is what we have and how it could fit. We've got challenges of how we might, operationalize that, but conceptually, that's the kind of structure that people's research is accommodating. So we do need to think about this, I think, from the publishing aspect and the kind of data that they would want or could potentially share. We then need to think about what kind of use cases we'd want to support for accessing this data. And we haven't done that exercise thoroughly yet, but now we have a model, we can start thinking about those questions. And I'd invite everybody to think about those kind of problems. And then we need to think about, what should an aggregator like GBIF do? What should others do? And at what level of complexity should it be simplified as it goes up the chain from the raw data up to sort of integration and use? We need to think through those problems. But we've now got it to a stage that we think there is a model to discuss those things. And that's largely why we're here presenting that to you today. So just to recap there, that's to acknowledge we haven't gone through that process of thinking about how someone might access and do questions that integrate all of this data. But if we were to have that sort of conceptual model of structure, we feel that we would be able to ask a lot of interesting questions here.
Over here, there's one over here too.
Sorry, I didn't realize we had to.
That's okay. Hi, my name's Jane Anderson. I'm from Local Context, and I just wanted to ask both, thank you for the presentations and for the case studies. I wanted to ask for the case studies, how you might have been dealing with Indigenous data. I'm just pulling a thread through from the keynote this morning. And then this possibility of the data package opening up space for Indigenous interests to be included. I just wonder how you're thinking about that as well.
Very good question. The first and simplest answer is that we haven't gone through a proper use case. And so there isn't a proper solution to all of the concerns that come up. However, there are working groups in TADWIG that are interested in these things. One of which is sensitive data that can be can cross the lines here with the same kinds of concerns that you're talking about. So as Darwin Core Maintenance Group, which is kind of responsible for all of this, we're aware of and trying to make sure that the solutions that come up there end up in the data package as well. And it's middle stages in terms of determining how that works, and also about integrating the care principles, for example, into usage policy. No, I would never claim that usage policy as it stands in Darwin Core Data Package solves the kinds of issues that will arise there, but it needs to. And as all Darwin Core things, it's a standard that's meant to evolve.
An online question from Jurid Pullen. I'm going to paraphrase a little bit. Sorry, Jurid. He wants something clarified. He asks, it seems like we suggest that Darwin Core Data Package introduces a way to record organism interactions, but that is also possible in Darwin Core Archives. Can you confirm that it is possible in Darwin Core Archives through resource relationship associated organisms? And Katie Pearson is backing this up also with phenological data. I mean, this can be represented with the extended measurement or facts, like why switching to Darwin Core data packages, and also can you confirm that organisms interactions can be well represented in Darwin Core Archives?
First, what was the second example? I got the whole thing about organism interactions, but there were phenological data. Okay, I'll deal with organism interactions. The answer is yes. The exercise to share organism interactions via Darwin Core Archives has been done in the past, and there is I don't remember, it's not called organism interactions, but interactions interest group that has gone through this as well. So yes, it is capable of doing it. However, like the OBIS extended measurements or facts extension to Darwin Core, it's a community understanding about a trick to do in order to make your data fit in a Darwin Core archive. It's not particularly semantically rigorous. It's hard to recognize an organism interaction from a material relationship because it's just so vague. A resource relationship is the place to put anything that you couldn't relate in the model before. So knowing that organism interactions are an important thing, we have a standards-based place to put them now. So yes, it worked before, but you had to understand how. You had to be in the in group. This will be part of a standard and documented.
Another online question from Javier Molina. He's asking, why is Darwin Core Data Package not using JSON for the EML? Seems a bit odd to have a data package JSON and EML XML. Why not use JSON for both?
Hi, Javier. Thank you for the question. It was actually to avoid changing everything all in one go, really. EML is an established standard. It's been used by our community in GBIF for 15 or more years. And we didn't want to tackle rewriting EML at the same time as this. I would call attention to the Catalogue of Life data package, which took the approach, I believe, of using Markdown for the metadata and translation tools to go from Markdown to EML. The metadata about the data set is not really the focus of our research at the moment. So the data set descriptive stuff that's in EML is not really what we're looking at with the schemas of the data package. I hope that answers the question.
We have one more virtual question online, and then I think we'll have time for one more question in-house before the session is over.
Okay. Caitlin Thorne is asking, will there be any Darwin Core Data Package introduction workshops?
Already missed it. It was part of the global nodes training. There won't be any in the rest of this unless you find me in a corner and keep me there.
Sorry, Deb, there's been a question identified already. Sorry. Ha, Deb, sorry. Oh, harsh.
Hi, John. Hi, Tim. This is Chandra. I work at the National Ecological Observatory Network in the USA. And I'm wondering if you could talk a little bit when you were thinking about how to create the data package, how you thought about that in terms of how it would interact with extended specimen infrastructure. And that can either be from, I guess, the model point of view or the front end point of view as well.
Hi, Chandra. Thank you very much. The extended specimen model, and there's actually many people in the room who've been heavily involved in that, have been collaborating with us throughout this process. So in the second session, there will be a presentation recorded from Sam from DISCO in Europe. Sam unfortunately couldn't travel, but his presentation is recorded, where the open DS specification or standard that they came up with, he demonstrates that he was able to map it faithfully onto the Darwin Core Data Package. And actually, he's someone who said it was actually quite easy. And it was easy because all the target entities existed and he could just transfer, well, I've got material, it goes into material. Got the collection event, it goes on to collection event. Got the assertions, they go, and for him, because the data structure was already largely aligned, it was a fairly easy data transfer. I don't want to give away the highlights from this presentation, but I would encourage you to come and see that example later. Thank you.
Okay, Deb, we have time for one more question.
Question, it's a comment to add on to what was said earlier. I think the point you were pointing out, Tim and John, the question that was asked about whether we should use the data package as it currently stands or the new one, I can remember, many of you can, I think we're evolving. Many of us are still in collection management software, for example, where we're just beginning to be able to get richer data out using the extensions that currently exist, right? We're getting there. I can remember asking Tim or Marcus at GBIF and saying, which one of your data sets actually use the extensions? Where are they? Wait, I can't find them, right? And then it's nice now in the user interface, right? We can see them. So this notion of how do we see the extended data that's there? And I think the new user interfaces that we're experiencing that begin to show us biological associations and relationships, we will see, we'll still have rows and columns of data, but we're going to have much richer visualizations of these data. I think that's where I see that going based on the history of what we've seen. So I think it's very exciting. Yes.
Awesome. Thank you very much, Deb. And that is time for this session. Remember, this is part one. It is time for coffee, but before you go, I would really like you to, like us all, to give a big round of applause to our presenters. Thank you so much for presenting your work. I know it took a lot.